In this last tiny sliver of the biology of learning and memory, I'm going to talk about the changes in the pieces, so the changes in the cells themselves, as well as the changes in the wiring between those neurons as we are learning new information. So what we're talking about with Hebbian learning is really long-term potentiation. So these long-term changes that are occurring in the relationship between the presynaptic neuron and the postsynaptic neuron as one or more axons are bombarding a particular dendrite with stimulation. And so that postsynaptic neuron is going to fire. And I'm going to do this in this simple way, and then I'm going to keep adding pieces to it. So if we look at the presynaptic neuron, it is releasing neurotransmitter, and then if that's getting the postsynaptic neuron to fire, it's going to be firing rather slowly with the first presentation of the stimulus. So this kind of fire, 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 fire. And as we get those neurons that fire together, wire together, as long as we are actually communicating directly, we see that there are changes. So in B, as continued presentation of the stimulus occurs, we're seeing changes in the presynaptic neuron. You can see that there is more area for neurotransmitter to be released. There's more neurotransmitter ready to go, basically, in that presynaptic neuron. And then also we see changes in the postsynaptic neuron, where we're starting to see some this is the building of receptors and the changing slightly of those dendrites. And we've looked at a picture before of showing dendritic spines that we can actually pull, create spines in areas off this dendrite so that we have even more areas that receptors can go so that we can respond to in a strong synapse, so we can respond more efficiently. And so as we're seeing these structural changes later, that same stimulus is presented again, and what we see is much more neurotransmitter release from the presynaptic neuron, more places, more surface area, and more receptors for those neurotransmitters to affect the postsynaptic neuron. So now, as we're getting that same stimulus as we did in A, instead of this slow kind of fire, 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 that postsynaptic neuron is going to be going fire, 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 fire very quickly. So long-term potentiation shows three basic properties that make it an excellent candidate for how we do this kind of learning, how cellular change can help us to learn and remember. And the first of these is specificity. So it's going to be only the active synapses that are strengthened. And in fact, if we see those synapses that are not being used, we're going to see a long-term depression. Okay, as we're going to start to that synapse is going to be weakened, and we might even prune that altogether in our synaptic pruning to make this a more efficient system. We also see cooperativity. Two or more axons simultaneously stimulating a postsynaptic neuron, and then that postsynaptic neuron firing, that causes stronger long-term potentiation than, say, a single axon in that kind of temporal summation, this kind of spatial summation with two or more axons. That's going to have stronger long-term potentiation. We also see associativity, which comes from these clearly. So pairing a weak stimulus with a stronger stimulus enhances later response to the weak input. Okay, and so I'm going to look at this picture with the neurons over there, and it's kind of hard to see for me on my screen. I know when I project it, it's large and it's easy to see. But if you look at the top axon, that's axon 1, and underneath that it says strong stimulation. And so we're just going to pretend, to make this easy, that the sensory neuron is axon 1, and the neuron that we're looking at, where all those neurons are communicating with its dendrites, that we're gonna call that the postsynaptic neuron. We'll pretend that's a motor neuron. And so what's going to happen, let's say with the strong stimulation, puff of air to my eye, that's going to cause me to blink. It's going to cause that motor response. And so that's going to be a very strong synapse. Let's just say I'm pairing a tone with the puff of air. So axon 2 underneath axon 1 there is axon 2, and underneath that it says weak stimulation. So let's just say there's a tone. Okay, and let's just say before we pair our tone with a puff of air to the eye, we play a tone. The tone is going to not have a very strong synapse with a motor response to blink my eyes, right? maybe I have a little startle response, but usually a tone plays, boop, and I'm not going to do anything in response to that tone. But we're pairing these together, right? We're getting the boop and then the puff of air to the eye. And so now we're seeing two neurons, axon 1 and axon 2. Those two neurons are communicating to the postsynaptic neuron, and we're going to have a really strong response because I'm getting a puff of air to the eye and I'm going to blink my eyes. And so now that specificity, axon 1 and axon 2, are both synapses that are being used. They are active, and they are both strengthened. Because of the cooperativity, this is causing stronger long-term potentiation, and because of the associativity, we're going to see stronger responses to that weak input, to that tone, so that pretty soon that tone becomes a conditioned stimulus, and I am going to just blink my eyes to the boop because it's been paired with the puff of air enough times. And we can also talk here about long-term depression, because if we look over to your farther right there, axon 3 is showing there's no stimulation there. And so that is a synapse that is not active during this whole process of learning that the tone predicts the puff of air to my eye, and that synapse is going to be weakened and potentially eventually pruned so that we can make this a more efficient system. The other thing that Calit is showing in this figure, in this diagram here, is the figure at the bottom, which is showing time and how the baseline response of the postsynaptic neuron to the tone is very low. So this is to my axon 2, to my weak stimulation or my weak synapse there. And then as we are getting the two pieces of information together, so axon 1 and axon 2 are both stimulating the postsynaptic neuron. During that dark blue bar, we see this great raise in the amount of response that the postsynaptic neuron is now going to have to that to axon 2, which before had no response, and now my response is going to stay much higher. Okay, we're going to look at some of the biochemical mechanisms of long-term potentiation, because the question is, now we see how long-term potentiation could really help us to learn, but how does that actually occur? And I'm going to say mostly this is due to glutamate. We're going to be talking about glutamate here for a while. We're going to go back to some of the really early lectures on how neurons communicate and what's going on there. And so if you are not remembering that, you might want to go back and look at that before listening or after listening, and then listen again, because it is kind of complicated, where what's happening with the glutamate receptors, as we've talked about with several other different neurotransmitters, but relatively briefly, and really just alluded to how we have these different kinds of GABA, we have these different kinds of acetylcholine, different kinds of dopamine, and different kinds of serotonin, and they each get their different names, usually with either numbers or letters after the D1, D2 for dopamine. But for glutamate, they are named after the drugs that we discovered that influence these receptors. And so AMPA, or AMPA, I usually call them AMPA receptors, are excited by glutamate and by a drug called alpha amino 3 hydroxy 5 methyl 4 isoxazolopropionic acid. I try not to say that one as much as I can possibly not say that one, but I usually say it once a semester and that's it. From now on, AMPA. We also have NMDA receptors that are excited by glutamate and by a drug called N-methyl D-aspartate. So these people named their drug much more reasonably, in my opinion. But I'm going to call them NMDA receptors. And what's happening, so if we look at the picture that Calit gives us of the presynaptic neuron and the postsynaptic neuron, the axon of the presynaptic neuron is releasing glutamate, that widely distributed excitatory neurotransmitter. It is fitting into the AMPA receptors, okay, and that's allowing sodium to enter. So it is an excitatory neurotransmitter. If you remember, sodium is a positively charged ion, and so in that that enters the cell through this ligand-gated channel that depolarizes the cell and raises its activation, its electrical potential. If we then look at what's happening with the NMDA receptor, the glutamate is, it would fit into the NMDA receptor, but there is a magnesium ion blocking that channel, and that's a positively charged magnesium ion, and it's going to be attracted to the negativity, right, the negative polarity within the cell. So it's going to be stuck in there, blocking the glutamate from getting into the NMDA receptor. So right now we're just allowing in sodium through the AMPA receptor. As we get enough activation, enough sodium and enough activation of the AMPA receptors so that the cell is depolarized, okay, that magnesium pops out of the NMDA receptor because it's no longer attracted to the negative polarity because it's no longer negative, and glutamate can activate the NMDA receptor, and the NMDA receptor also allows in sodium, but it also allows in calcium, which is also a positively charged ion. But calcium does a number of things. It has some it allows for metabolic changes within a cell, and so now we can activate a protein which sets a series of events into motion where we can, more AMPA receptors are built and dendritic branching is increased. And so as we just saw, earlier in about three or four slides ago, we just saw these changes can now increase the later responsiveness of the dendrite to incoming glutamate. Okay, so I've now strengthened that synapse by both adding AMPA receptors and more dendrites by allowing in the calcium through the NMDA receptor. And those are really the mechanisms of long-term potentiation. We also do see changes in the presynaptic neuron as well, as extensive stimulation of a postsynaptic neuron is going to cause the release of a retrograde transmitter. That nitric oxide is going to travel back to the presynaptic cell. If you remember, that fit in to tell the presynaptic cell that, hey, you've sent enough neurotransmitter, but what it's also doing is it's allowing for some of the following changes. So a lowering of the action potential threshold. So I can now, as the presynaptic neuron, I can now respond with less depolarization, an increase in neurotransmitter release, an expansion of the axons, which is what we saw in that really early long-term potentiation slide in that picture. And then it can also release neurotransmitter from additional sites. And so again, now strengthening that synapse. We just talked about what's happening at the postsynaptic neuron. This is what's happening at the presynaptic neuron. Both of those neurons, we can see cellular change in them so that it's changing the wiring between the neurons, or the communication between the neurons. Long-term potentiation is important for certain kinds of learning. We see many of these kinds of receptors in the hippocampus and the amygdala, and places where we see the kind of learning that we've just been talking about. But over time, there does appear to be a shift to the cerebral cortex, and really we're storing information in the cerebral cortex. And that shift is not very well understood at this point. Okay, so he ends this section of learning and memory discussing improving memory and how we can do that. And as many biological textbooks do, he discusses drugs and some of those transcranial magnetic stimulation, or how we can excite neurons, which some of that is so new and so questionable as to the long-term effects. But even with the drugs, we're going to say it's pretty questionable as to the long-term effects of really improving our memory. He starts with caffeine, Ritalin, methylphenidate, other stimulants. The Ritalin and the ADHD drugs that people are sometimes being prescribed and then selling to others, especially in college, because people want this ability to pay attention and be aroused while they are studying for an exam. Those are really short-term effects. And caffeine as well, there appears to be a plateau with caffeine where we can really be taking away our ability to have focused attention if we are drinking too much caffeine, and it does not make up for just sleeping well. As I think at the very end of this chapter, he says the best thing to do is to get exercise, eat right, and sleep well. So those are really important things, really, for improving memory. And again, those drugs that he's discussed are really short term. What I'm going to do right now as I'm studying, and it is not clear whether that means you're going to have better memory for that material sometime later in the future, and what the long-term effects are. And in fact, many people are abusing some of those drugs. He discusses here ginkgo biloba and says that the research on this is really questionable. I will agree with that. Ginkgo biloba, first of all, is something that you would be taking for months before you even considered maybe seeing an improvement of memory, and it appears to be pretty subtle. I have looked into this a bit, as one of the books I've read about improving memory he does suggest ginkgo biloba and improving a lot about our brain. He suggests taking ginkgo biloba. What I've noticed about the people who are recommending ginkgo biloba is kind of what I've noticed, honestly, in the meditation research. I am very pro meditation and pro ginkgo biloba, but when I go and look up the research, there are people finding that these things help in whatever we're interested in, and I'm going to stick with the ginkgo biloba and memory, and there are people who say there's no influence at all. And the people who say these things help seem to have a vested interest in finding that outcome. And so the research is really questionable, in my opinion. But I take ginkgo biloba because I would do anything, as I've already discussed the Alzheimer's in my family, and you might be able to tell that my memory is really important to me, and I'm 50 years old, and I used to have almost an eidetic memory, and so now I have this. I can see where my memory lapses as I'm aging, and I hate it. That's the thing I never wanted to lose, as I had an excellent memory. Another herb that he discusses in the book is called Bacopa monnieri, and I don't have that on the slide because it wasn't something that I had heard of or looked up at all, but this is a water hyssop, and this is something that has been used in India in Indian medicine since the is it the 700s AD, for improving memory and attentional skills. This is another, I'm not sure what the research is on this. It's kind of questionable, and it's another one of those herbs that it's going to take more like a month or months to see any influence of that at all. But from what I've been watching now, because I've never looked this one up, it does appear to have those antioxidant effects, and because that is so important for fighting cancers as well, and because antioxidants are so good for the brain, I'm going to probably go look into that quite a bit more. I think I know he ends by saying the best things to do for your memory are eat well, exercise, and sleep well. I know aerobic exercise, we see an improvement in memory, and we're going to talk about that one more time when we talk about how exercise helps depression, that we're seeing the same kind of boost in the neurons, in the neurogenesis in the hippocampus, which is really important for memory. But another really good thing to do to improve our memory is to study really well in the first place and develop the strategies that help us the most. I'm not going to say things against flashcards. A lot of people use flashcards. I never used flashcards, but as I just said, I never studied a great deal, as my memory was just really, really quite good. But flashcards are this real, if I want to remember this for the test tomorrow, I'm going to just memorize some definitional things. Whereas what we want to do when we're really learning, when we're really building the semantic space, first of all, we want to build the semantic space so that when we learn something new, it fits nicely into that semantic space, and we're more likely to remember it. But one of the ways to do this is to really develop questions. So I have had one book that suggests that you go and write the chapter headings and the subchapter, all of those little headings, write them out Just so if I go and look at a couple of these, I would say AMPA, NMDA synapses, presynaptic changes, improving memory, and develop curiosity. So maybe write out a couple of questions for yourself that you might have. And what's even better is to give yourself some potential answers for those questions and then go and read. And because what we're doing when we're developing curiosity is we are seeing activity across more of the brain than when we are just mindlessly kind of passively reading. And when we come up with a question, we come up with an answer, and then we go and we find out that that answer was wrong, and we hear the right answer, we lay down those memories the best. That is one of the reasons I partly give the test corrections to help my students, because sometimes the exams are really hard, and it's really hard material, and you just having that second chance to come back and look up information can help your grade, but more it is to improve your memory. And I feel like some of those students who don't do so well the first time and go back and find those answers, they're more likely to remember those answers, the ones that they got wrong and had to go look up the correct answer, than anything else on the exam. So these are the ways really to improve your memory, is by finding that curiosity, which helps with all of that arousal and attentional and all of those pieces and parts that play into laying down a new memory. All right, that is my end of learning and memory. I know I've gone on more than I wanted to, and so at this point, I'm going to skip cognition. We have done pieces and slices of this throughout perception, throughout when we talked about conscious versus unconscious, and more recently in learning and memory as well. There has been overlap, and I've taken parts of this chapter, so the only thing I'm really missing in cognition is some of the basics behind language. I'm a language person. It bothers me a little bit, but I know it's time to move on to psychological disorders so that we can get through the psychological disorders. That's usually what students are very interested in. So we're going to start talking today about either schizophrenia or depression. I'll have to go see.